Okay, good morning, everybody. Thank you for this incredible opportunity to tell you about eCLOSE. It's a new nonprofit research institute that aims to empower the community lens on science through education. So before we get started, come on. Okay, click here. Before we get started, I just wanted to invite everybody to go to joinpd.com and type this code here. We're going to have a few interactive slides in a couple of minutes, and it would be wonderful if everybody could participate. So again, you just go to joinpd.com on a different tab or on your phone, and then type in this code, AOUNNH. So my research lab is interested in identifying nutrients that impact signal transduction to affect stem cell lifespan. This is a current view of the daily recommendations from the United States FDA, which really focuses on sort of broad strokes of encouraging people to eat more vegetable-based products and reduce the processed food and protein and high fat. The impact of diet on the process of aging is unambiguous at this point, with reduced calories and starve feed cycles really affecting the stem cells to reduce the effects of aging, suggesting that understanding the precise molecular effects of individual nutrients might actually enable us to enhance this aging benefit without really having to make people starve. So several years ago, we identified cholesterol as a trigger of hedgehog signaling in the fly ovary, defining the stepwise signal transduction pathway that releases the hedgehog molecule to affect proliferation of epithelial follicle stem cells shown here. We went on to show that the balance of cholesterol and dietary protein and sugar impact hedgehog signaling, which remains continuous throughout the lifespan, versus insulin signaling, which reduces around middle age. Continuous hedgehog signaling promotes induction of autophagy and loss of stem cells from the niche, effect that is counterbalanced by insulin signaling up until the middle age. And so this suggests that the precise balance of individual nutrients that affect single signal transduction pathways can have a profound effect, both on an individual cell and also on the function of a whole organ. So from this, at the same time as we were setting up our large outreach program, we developed the hypothesis that everything you eat, every chemical that goes into your body as part of your diet, affects a signal transduction pathway, with the collective response determining the outcome on an organ. So our goal now is to map individual nutrients onto this beautiful protein interaction map in Drosophila, such that we understand how each and every nutrient affects every one of these dots. So a daunting task, but once we figure out what the precise molecular impact is of each and every nutrient, we can start to tailor personalized diets to improve targeted therapies for cancer and other diseases. So now back to joinpd.com and type in your little code here. What you should be seeing on your Pear Deck screen is an option to vote here, whether grapes are healthy or not healthy. So we need to know where to start in terms of our nutrient screening, and we're looking for your help. So if you can type in whether you think grapes are healthy or not healthy, we'll see what we think. So in general, most people think that grapes are pretty healthy. I'm guessing the not healthy is maybe because of high sugar. Okay, so whoops. How about candy? Healthy or not healthy? Healthy, that's funny. Okay, and that gets maybe a little bit more confusing. What do you think about eggs? So yeah, when I first started doing this, most people would say that eggs were unhealthy, and then the dietary recommendations changed. And so one of the things I really want to emphasize is it sort of depends who you are. So eggs are very healthy for people because of their high mineral content and vitamins. But if you have diabetes, heart disease, high cholesterol, high blood pressure, metabolic disease, eggs are really not your go-to food because the level of cholesterol is much too high and can actually be dangerous. So this is the last one, orange juice. Right, so here the nuance becomes clear, right? Orange juice is healthy. It has vitamin C, it has all kinds of fiber, you know, in the juice. So many of the healthy things that we need to keep our bodies running, particularly vitamin C. But for cancer patients, it's on the do not eat list. Orange juice counteracts the effects of chemotherapy, and therefore is not something that you want to take depending on your condition. And so this quiz is supposed to emphasize the nuance of what happens, you know, when you eat something that is considered generally healthy, but you have an underlying condition that may make it not healthy for you. So what we want to understand is the molecular mechanisms that determine whether each individual nutrient is healthy or not healthy. And so what we really would like to do is to start to leverage cultural experiences of people everywhere, their religious cuisines, their cultural cuisines, things that they use to try to promote health. And so we have in our fly community here people from everywhere. And so what I really like you to emphasize on here, which may make all of these little things move quickly, is what is your cultural cuisine, right? So I live in Pennsylvania. I grew up in New Jersey, New York area, but my cultural cuisine is probably not that, right? So yeah, so right, and now all of a sudden the bars start to move. We have people from all over the world with different ideas of what is healthy and what is not healthy. So in the next slide, you're going to type in what you think is healthy and not healthy to give us some ideas of what we should start to investigate. We really have focused mostly on foods that are prevalent in the United States, and our students, as I'll show you in a minute, have brought in great, fantastic ideas from other places. So, okay, I don't know why we're only seeing the first ones. It should be scrolling, but anyway, got to work on this one. So yeah, so basically the idea for everybody is that every single person eats, right? Everyone eats. Everyone has an idea of what foods are healthy, what foods are not healthy, including children. So what we've tried to do is to leverage the life experiences of people ranging from 5th grade all the way through retirement to try and identify nutrient compounds and diseases and to pair them together to try to start to tackle the health conditions and the health challenges that face individual communities using the people who are in that community to do the work. And so these are our high school students, and these are summer campers. We have our teachers like bent over in severe concentration to learn how to do the research. And we engage the families and the communities of all of our participants in the work. So our goal is to teach kids to conquer cancer. And as I mentioned before, when you saw that incredible protein interaction map, this is a daunting problem. So even for cancer, which is where we started with this, there are 1 million different nutrients and 300 cancer genes, making a single combination of 300 million nutrients. But luckily, there are many hundreds of millions of high school students worldwide. And then as we expand out to include everyone in the research, it's almost an unlimited pool of scientists who can generate data to contribute to this project. So how do we do this? We built the program on the concept of citizen science, which starts in level 1 as crowdsourcing, moves on to distributed intelligence, participatory science, and then extreme collaborative science. So the way we do this is we start with a project called Flybox, which many of you will be doing in the eCLOSE workshop next Wednesday, where we have the students bring in what they think are healthy and junk foods and test the effects on the fly life cycle of foods that they consume in their regular life. In the classrooms, in partnership with teachers now all over the country, we move on to test to include more ideas from the students. The classrooms will pick a disease and a series of nutrients that they are most interested in to investigate as a group. focusing on signal transduction. In the summer camps, it starts to become more independent with students working on individual ideas and evaluating their data to contribute back to the program. And in the undergraduate bridge level, the students actually drive the entire project. So they come up with the question, they come up with the hypothesis, they develop an experimental plan and execute the experiments and collect the data. And then the students who excel in this undergraduate bridge program move on to what has been our CURE program from NCI for many years, where they join research labs at Fox Chase and at Penn Medicine to basically leverage all of the projects that they've developed here to make a contribution to an independent research lab. And by the time they hit this platform, they've just finished high school, right? So they haven't even begun college while having all of this research experience under their belt, putting them in an extremely strong position to succeed in STEM majors, lab research, and then move on to their future careers. So our goal is to empower students, especially in Philadelphia. And this is what we find as a very sharp contrast between the demographic composition of US faculty research scientists by race compared to the demographics of the school district of Philadelphia. And you can see it's inverted, right? The predominant populations in the school district of Philadelphia are underrepresented minorities, where the predominant population of faculty research scientists are white males. And so we see this both as a really striking image of the challenges faced and the marginalization of students in inner city schools, as well as a powerful opportunity to address the health disparities that are experienced in these same communities, with highlighted most recently by a threefold higher incidence in many zip codes in Philadelphia of COVID-19. So what exactly do? We take fruit flies, which we all love. We feed them so far mostly with dietary supplements, junk foods, or healthy foods, and then evaluate the impact on the life cycle. We found that the most robust quantitative assay is counting pupil, which can be compared between students doing the same data point, and also to evaluate the effectiveness of the program in classrooms. So the beauty of fruit flies is that there are flies that bear oncogenes and tumor suppressor mutations that allow us to compare the impact of individual nutrients on whole flies. So we use homozygous viable mutants, whole flies impact between wild type and mutant. And as we know, scientists have developed models for pretty much every human disease. So when we're ready to expand, and when some of you say, hey, I want a piece of this, we will be able to expand into additional diseases, depending on what is most important to the community. So this is the 2019 presentation day for our classroom program, where 570 of our 1,000 students presented their work to hundreds of scientists at the Franklin Institute. It was an incredible day, and each year now all throughout Philadelphia, we have high school teachers joining our group to impact so far over 2,000 students. And this spring, we anticipate having hybrid at-home participation for somewhere between 500 and 800 students. The students are generating powerful data sets, and we are certainly having a tremendous impact on the students of the city of Philadelphia, who I mentioned before are largely underrepresented minorities. So does it actually work? And in sort of a flipped idea, we usually try to emphasize the positive. I just want to point out that the blue and the red are students who reported that they had no gains or just a little gain. And you can see in the pie charts for life skills like peer collaboration, confidence, adjusting to projects, thinking outside the box, the blue and the red are teeny, like 80 to 90% of students reported high gains in these, just from this one little classroom program. Similar for research skills, inclusion, and interest in research. And then finally, we were very interested in finding out whether this program distributed to unselected students, not in science, not determined to be scientists. Do you want to be a scientist? And 75% said maybe or yes, with an even higher number saying they wanted to do research, which I think is is an incredibly interesting thing we could talk about more. So the students can then move on to summer camps, which again started as a face-to-face and moved to a virtual format last summer because of COVID-19, where students received research stations in the mail and did the exact same program that the face-to-face students did, but working at home. And one of the most important parts of our program is representation. So our instructors and staff mirror the demographics of the student participants to ensure that there are enough cultural awareness and also role models for the students. And so, as probably expected, with more engagement, more focused attention, there was even more interest, where all of the student participants thought it might be possible for them to be a scientist, and 90% are interested in pursuing more research. So our level 3, which is actually where we started in 2013, is the undergraduate bridge program, where students focus on signal transduction. And in recent years, students have identified grapeseed and burdock root as specific inhibitors of particular signal transduction molecules that are highly important in cancer and diabetes. And perhaps more importantly, they've identified compounds that bypass targeted therapies. So selenomethionine and black cohosh here activate the same protein, which drives activity of the RAS signaling pathway, even when EGF receptor or insulin receptor are inhibited. And so we're hoping to leverage these discoveries by the students to develop elimination diets. So don't eat selenomethionine if you're on Herceptin targeting EGF receptor types of therapy. So some of the students who come to the undergraduate bridge program bring their own projects, and this is 2 examples with high impact on public health. One student came with a bag of apricot seeds saying, my mom and all her friends who are breast cancer survivors are eating 17 of these a day. And so, okay, that's really interesting. Let's figure out what's going on. The apricot seeds contain a compound called amygdalin that breaks down into cyanide and benzaldehyde, with the highest likelihood of the patients getting cyanide poisoning rather than having any effect whatsoever on suppressing their breast cancer. Another student came in saying, I really want to study weevils, which in her grandpa, who had stage 4 lung cancer, was eating 100 of these live a day in a glass of Sprite. And this is widely used throughout South American communities to combat cancer, diabetes, allergy, asthma, pretty much anything you can think of. And so is there anything to this, or is it just like you're eating live bugs? And so it turns out the beetles secrete a defense chemical that may have chemotherapeutic properties, something that we're going to be investigating further, while at the same time trying to figure out how to get the public health message that ingestion of these bugs can cause anaphylaxis, and also the carcasses aren't cleared from the intestinal tract, causing inflammatory bowel disease. So these are, I mean, what I'm trying to say is, like, there is no way that you're, trained, went to grad school, did the postdoc scientist is going to hit on these types of questions as a major focus. And, the undergraduate bridge is so phenomenal. All of the students want to do more research. And again, note that the same students who answered the survey, not all of them are sure they want to be a scientist. So the research is what is really driving their excitement and interest. So what has turned out to be the case is that we're kind of flipping the statistics on success rates for STEM, where nationwide 77% of college students switch out of STEM majors, but 72% of our graduates continue doing paid research in college. In Philadelphia, fewer than 6% of students complete STEM degrees, with 10% fewer even entering graduate degrees, let alone completing them. But 100% of our students so far have completed their STEM degrees on time. So the biggest mark of success is true independence. And this project blows my mind. So these 5 women got together to create a project focused on HPV prevention based on a cultural awareness project that Ileana here did a couple of years ago, where she found that the types of HPV oncogenic strains that infect Latina women were different than those in the majority white women. the pink here shows the strains that are covered by current available vaccinations. And here you can see that African-American women have oncogenic strains of HPV virus that are not covered by the vaccine. So Nicole here decided to investigate mistrust among Black communities for getting HPV vaccination and wrote a paper. She did this project and wrote a paper in less than 6 months because of this intense passion for the problem and understanding how these students can actually educate and help their communities to make decisions about their children's health. And then Nilise, Allison, and Jesse here focused on the molecular impact of the HPV strains with the goal of identifying nutrients that might kill cells with low P53 and low RB, which are the proteins targeted by HPV, before they actually transform. And Jesse focusing on the molecular biochemical differences between the HPV strains. So this is a project that we're going to be expanding. You can already probably guess the worldwide implications of how powerful the results of their planned project could be. And these women will be at the helm. And the most important part of this is the oldest of these women is currently a sophomore in college. So the energy that drives the progress of eCLOSE and previously immersion science is the community. And so you can see here that students from our undergraduate bridge programs are from all walks of life, all cultural backgrounds and all income levels who come together to know each other, truly know each other as individuals with strengths and weaknesses to drive forward the scientific discoveries. So I wanted to sort of end on 5 lessons that we've learned from doing a program that has exploded in ways that we never, ever anticipated. And the first one, most of them are suggestions for ways that PIs can actually continue to promote the success of these students. So the first is to take responsibility. This is an image taken from a recent Time magazine article describing how in C-suites and businesses, generally the only underrepresented minority people sitting at the leadership table are the diversity officers who face this tremendous uphill challenge to discuss issues and interventions that can improve climates and environments, while at the upper end of the table, you know, these people think they're kind of reaching out to save somebody, do you a favor by giving you a job, and school you on what the company culture actually is. And so what I want everybody here to think about is, does my lab meeting look like this? Is this what my graduate school classes look like? What about thesis committees? Right? Are we trying to save somebody, or are we trying to engage the vast life experience and interests and capabilities of diverse people to enhance our own work? And so at the end of this meeting, you can imagine somebody at this end says, oh, you know, I think we all ordered pretty much the same thing. Let's split the check. And then everybody from here down is ready to puke because they only have $4 in their wallet after sending their money home to support their families. So I think we really have to start to realize in images like this that there are disparities going on in our everyday life that can make it like nearly impossible for the people at this end of the table to actually start to move up and feel included and contributory. So we have some suggestions, which is pay for your lab social events. Don't leave anybody out, particularly the students who say they always have homework to do, because they probably are saying that because they can't afford to be there. Other things that have really worked well for us is have a cultural cuisine potluck for your lab party, where every single person can contribute and open the doors to those kinds of conversations about culture in a completely contributory way. If you can avoid the only, like have one person in your lab who's completely different than everybody else, while that enhances diversity, it's very lonely and challenging. Take the time to understand, especially the strengths of the diverse students in your lab and environment, and take the time to bring up the weaknesses that they may have experienced due to a subpar education that has nothing to do with their capabilities. So the second one is ensure safety, which is sort of a corollary of the responsibility. it is your responsibility, it's our responsibility to be the shield between institutional biases that negatively impact our students, to foster those open conversations where you can discuss, like, how do we all feel after these tragedies that have been going on in our country? How does it impact our relationships with each other? And have those conversations openly in lab meeting. If you are unsure what to do, don't hesitate to involve the experts. Every university has them, and they are thrilled to be able to help. And finally, the most important thing is to speak up. If you're walking with your graduate student or postdoc and somebody's looking sideways at them or suspicious, or they come and tell you, you know, somebody asked me for my ID when I was walking to go, you know, get my mice or whatever, you got to speak up. You have to say, no, that's not okay. And I'm going to do what I can to be on your team. You can augment the scope of your work, leverage the diversity to include, in addition to the underlying genetics and biology, environment and lifestyle, based on the experience of diverse people who come into your life, to raise this up into a comprehensive study for how all of these factors and social determinants of health can impact biology. And you know, substantial recent work has shown that race is not linked to genetics, but diseases are, and diseases are both linked to genetics and to community effects. I already gave you one example, like HPV, where different strains affect different minority communities in particular. Same with BRCA gene mutations, which affect Ashkenazi Jewish populations, as well as is very high in Hispanic. And then prostate cancer is 60% more likely to occur in African-American men, and the drivers of this disease are different than they are in predominant white populations. Finally, there's a very terrifying rise in early onset colorectal cancer, which exhibits differences in geography such that minority populations experience left side colorectal cancer, which is harder to detect, versus right side, which is the more well-studied. And it seems to be due to epigenetic changes. And so you can really leverage the new emerging genetic information on these diseases together with environment and lifestyle experiences that maybe you have never had to develop unique questions and perspective and increase the impact of some of your work. So the next lesson is to celebrate individuality. So all of these students are classified as minority students by our society. And you would never know from looking at them which one cried every single week of our, of our program, fearing failure. You would never know which two are best friends now and college roommates. You wouldn't know which one basically transformed her entire high school by having such a positive experience with our program that now the entire school and every ninth grader in there participates in the research, or which student had beautiful, incredible, powerful ideas from her experience with her grandmother's breast cancer, which were dispelled by an outside, well-meaning faculty mentor who said, don't do it that way, do it my way, it's better. So what we have to really understand is that minority students are individuals just like everybody else. And if you discuss where they're coming from to understand, instead of dismissing their ideas, build from the foundation they're starting from. Don't refer to this group of young ladies as the minority students, right? Unless they request to be used that for their pronoun, and avoid assimilation. Don't say, you have to do it my way, like let's get rid of your way. Similarly, fostering interactions between students from different places has been incredibly powerful for us, as well as community effects, where this is Marcus and his 4-year-old sister, who he hired to be his scientific technician during our summer camp program. And this is her saying, when is it my turn to be a scientist? So these are the kinds of things you really want to do. And you can see here, like all of the students have an idea. Everyone wants to contribute, even that kid who sits all the way in the back trying to hide from you most of the time. So I want to end on what was a failure on our part. This This was a project presented in our undergraduate bridge last summer by a student named Jocelyn, who wanted to work on diffuse large B-cell lymphoma. She wanted to work on BCL3 and to understand whether there were dietary interventions that might improve this disease. Clearly, flies don't have B cells, and so we tried to redirect her onto understanding how BCL2 might affect apoptosis and whether there were nutrients that could enhance its efficacy in killing cells in general, since we couldn't work on B cells. And throughout the program, the instructors kept reporting back like she's just not engaged, she's not interested, we can't convince her to really, you know, dive into her project. And 2 days before the presentation day, we discovered that Jocelyn was diagnosed with diffuse large B-cell lymphoma last year. She's been on what she described as a roller coaster, one of the extremely rare patients who's between 15 and 24 with this disease. And in her case, it happens to be driven by BCL3. We missed the whole boat. We failed this student. We failed to recognize her courage and her fortitude in saying, I am going to do something about a disease that affects people like me, in the hope of preventing them from having her experience. And so what I want to say is we tend to tell students a lot, like, you have to learn how to accept failure. And I want to disagree with that emphatically and say we can't afford failure. Certainly not with students who are this vulnerable, who have so much to give and really need us to promote their success. And so this is the most important of the 5 principles we've learned, is always honor the purpose. Why is the student here? What is it they want to do? Don't redirect them into something. Just find a way or find a different lab that can actually honor their purpose. And so what we're doing is creating a hub where students, teachers, and scientists can come together who are going to take responsibility, ensure safety, augment the science, celebrate individuality, and honor the purpose, to generate tons of data and a network that will promote the student's success over time. And so I want to thank all of our funders, and notice GSA is on here, who funded 7 of our trainees to promote a career session at that giant Franklin Institute symposium a couple years ago. And we just received a Drosophila outreach grant to support scholarships for the summer camp programs. eCLOSE is built on friendship. So this is Dara Ruiz-Whalen, who will be running the workshop next Wednesday. We have been friends for a long time, and the core of energy from friendship is what we distribute throughout the entire program. We recruited Sybil Bryant to be our Chief Operating Officer, and we have a powerhouse team of advisory committee team. These are our instructors who've been with us for years and years, also all of our best friends. And this is Ebony, who taught me how to do the interactive pear deck. So here's our website here and our email address if you want to get involved, and I'll be happy to take questions.